Uh, Welcome, everyone. Thank you for tuning in today. Um, and welcome to this series of talks. This is the public and patient engagement series of talk of the British Society for Gene and Cell Therapy. The aim of these talks is to introduce to the public research being done in the gene therapy and cell therapy fields. The idea is that the public can access this information, understand the developments that are being done, hopefully ask questions to the researchers that are doing this research, these investigations, and just basically bring the science that people are currently developing to the general public. My name is Juan Antinao. I am a research fellow at UCL. I work together with Dr. Sonam Gurung and Dr. Rajvinder Karda organizing this series of talks. And today we have with us Dr. Karina Graham. She is a postdoctoral research fellow at King's College London. She did her undergrad in the US and then worked for 2 years in the US National Institute of Health, after which she moved to London to do her PhD. As I said, she currently works in King's College London as a postdoctoral research fellow, researching a gene therapy or a gene editing approach. She will tell us more about that in her talk for epidermolysis bullosa. And today she will talk to us about her research, what the disease and how her treatment approach would work for this disease. Her topic is called the Ikea model, why your cells are basically a DIY manufacturer, furniture manufacturer, and how we can use that to treat genetic diseases. So I will leave her to introduce her topic, and then if you have any questions, please write them on the YouTube comment section or in the live chat, and then we will read them out loud at the end of the talk so we can discuss them.
Okay. Thank you so much for the introduction. I hope everybody can see my slides. But yeah, I'm Karina. I'm a postdoctoral researcher, and today I am going to tell you a little bit about my research. But I first wanted to sort of just lay the groundwork for the way that I like to imagine genetic disease through this analogy that I call the IKEA model. So I think we're all familiar basically with how IKEA works. When you need something, you send away for it, and they send you the parts that you need to build it and an instruction manual that will tell you how to put everything together. But what's actually happening down at IKEA HQ is that they have to go into their big database that has all of the files for all of the objects that they know how to make. And within this big database, you can think of all of that collection of files for all of those instruction manuals as your DNA. And then each individual file for each individual instructions manual for each little piece of furniture, those are your genes. And so you start at the DNA level. And then as your orders come in, they will print out these little single-use instructional booklets for the specific thing you need to make. And then it's your job to sort of read that and put it together according to the instructions. And within your cells, this is a process called, and if there are any geneticists in the audience, you already know what I'm going to say, transcription, which is the act of sort of converting or copying the DNA into an instructional molecule called mRNA. And then to go from mRNA to your protein and sort of assemble the protein, this is a process called translation. And what you end up with is a functional protein. And proteins are essentially just equipment that your cells use in order to perform their jobs. But what happens when the file contains an error? So as you might imagine, if there's a typo in that original file, the instructions manual that gets printed out and sent to you is no good, and you're going to end up with a protein product that doesn't actually work the way it's meant to. And if your cells try to use that protein anyway, you're going to end up hurting yourself. And these are the symptoms of a genetic disease. So how genetic disease works: proteins are tools that cells use to do their jobs. Mutations in DNA are typos in the instructions to make those proteins. Those faulty tools means that the cell cannot function, and that's how you get these disease symptoms manifesting. So what do we do about this? So there are a couple different treatment options for genetic disease. And before we were capable or knew a whole lot about how the upstream side of it works with editing, the capability to edit DNA and mRNA, originally, it was a lot of small molecule pharmaceutical intervention. And essentially, these small molecules serve as extra pieces that will stabilize the chair as you're building it or after it's built. They kind of help nudge that protein into the proper conformation so that it will work as well as it ought to. There's also mRNA replacement, which is where you send a corrected manual alongside the faulty one, so one that doesn't have a typo in it. But within your cells, that manual sort of self-destructs after a single use. So you have to keep on providing your cells with this corrected mRNA. And then what I study is DNA editing. So you go into that master file at Ikea HQ, you correct the typo at the source, and it fixes the problem, in theory, permanently. So my research, as said, is on epidermolysis bullosa. Essentially, it's a dermatological disease, and your skin has a couple different layers to it. And the two main ones that are relevant in the disease that I work on are the epidermis, so the outermost layer, and the dermis, which exists underneath. And these two meet at what's called the dermal epidermal junction. And at the dermal epidermal junction, the thing that holds them together are called anchoring fibrils that are made up of protein. And the specific protein in the disease that I work on is called collagen 7, which is encoded by a gene called COL7A1. So if you have mutated COL7A1, you get no collagen 7 protein. No collagen 7 protein means you don't have any anchoring fibrils. And no anchoring fibrils means that you get formation of blisters. And over time, these blisters will grow until they rupture and become wounds. I'm going to show an image now of some patients with epidermolysis bullosa. There are very large scale wounds on them. So if you're squeamish at all, I would encourage you to look away, and then I can tell you when you're going to look back. So here we go. On the left, we see an infant with EB, and you can see that even very shortly after birth, these large-scale wounds start to appear because the dermis and the epidermis, those two top layers of your skin, are just not adhered to each other. And with any friction whatsoever, it will cause the shear stress that causes blister formation and wounds. And those wounds don't heal. They're constantly trying to heal, but they essentially can't keep up with the formation of these blisters. And as you might imagine, if your cells are constantly cranking out new copies of themselves, that leaves the door wide open to errors, which can lead to cancer. And so on the right, we see a patient's hands and feet with what's called squamous cell carcinoma, which is a very aggressive form of skin cancer that results from the sort of chronic inflammation and elevated healing state that patients with EB tend to be in. And squamous cell carcinoma tends to be the thing that is actually causing mortality in these patients. So the wounds are off the screen now, so you can look back. So one of the questions that we have to deal with when we're coming up with a DNA editing treatment is, do we want to go for an individual mutation correction, or do we want to go for a total gene replacement? So different DNA editing technologies have different mechanisms of action. You've probably heard of CRISPR before, and CRISPR can be divided up into different, I like to think of them as like flavors, and there's flavors of CRISPR that are really good for tweaking individual base pairs within your DNA, and then there's other flavors of CRISPR that are better for these large scale edits. And which one we use depends a lot on the nature of the disease. So for something like sickle cell anemia, every patient with sickle cell anemia has the same underlying mutation. So those individual correction flavors of CRISPR are the ones that have been used to develop therapies for that. But for epidermolysis bullosa, there are over 800 mutations that have been identified in these patients. So in this image, this long rectangle is a representation, a graphical representation of what the COL7A1 gene looks like. The different colors within the rectangle represent different pieces of the protein that help it do its different jobs. And each one of these little labels represents a mutation that is known to have occurred in an individual EB patient. So going in and individually correcting these mutations would, in theory, work for any one of these 800, but it's really difficult to get FDA approval for 800 different unique therapies. And I can talk a little bit more if anyone has any questions about the regulatory part. I do know not a whole lot, but a little bit about why that's so difficult and what's being done to try to address it. But as of right now, we would have to essentially get individual approvals for each one of these individual mutation correction approaches. So for epidermolysis bullosa, which is caused by 800 different mutations, it's just not really that viable. So what I was interested in and what I was hired to do is explore this DNA editing technology called PASSIGE. It's very new. It was published in June of 2024, so just about a year ago. And the title of this paper, if you're interested in learning more about it, is Efficient Site-Specific Integration of Large Genes in Mammalian Cells by Continuously Evolved Recombinases and Prime Editing. And in sort of simpler terms, site-specific integration, meaning that we know exactly where in the patient's DNA the copy of this large gene is going to go. Mammalian cells, obviously we're mammals, so that's good for us. And then the continually evolved recombinases and prime editing refers to the fact that the proteins involved in this machinery have been evolved specifically for this purpose. And again, I can talk a little bit more about how that technology arose if anyone is interested and wants to ask a question about it later. So how it works essentially is that you probably know that viruses, or some viruses, evolved to paste their DNA into host DNA. So it's kind of like a cuckoo bird laying its egg in another bird's nest and tricking that mother bird into raising that egg as its own. The virus will plunk its DNA down in our DNA, or whatever organism it evolved to invade, and trick that cell into cranking out more copies of that virus. But they can't do that just anywhere. They have to target these specific runway sequences. And DNA editing can be used to program those runway sequences into safe spots in human patients' DNA. And safe, meaning that extensive research has been done that shows that altering DNA at this point doesn't have any detectable negative effects on the cell. So this leaves us with the opportunity for a one-size-fits-all treatment for epidermolysis bullosa. So we use small-scale DNA editing that's very safe, efficient, and precise to make an edit in EB patients' DNA that builds that runway for those proteins isolated from a virus. And those viral proteins then deliver a healthy copy of the collagen 7 gene. So we take the viral DNA, the harmful part, remove it completely, and we hand it a healthy gene instead, or a gene that's going to help these patients become more healthy. And this gives the cells the ability to make their own collagen 7 permanently. So you don't have to take a medication for the rest of your life. In theory, if this works, you just one and done, and you're healthy forever. Not forever, but you know what I mean. So step one in my research then was proof of concept. And this is step one for basically all scientific research projects. So we started in kidney cells because kidney cells are known to be very receptive to delivery of genetic material and DNA editing. They don't mind their DNA being futzed with. So it's a really good way to optimize your approach. So what I did was I inserted an antibiotic resistance gene at our target safe harbor locus. And any cells that successfully had that gene integrated into their DNA could survive a 2-week course of these antibiotics. And you can see here that our non-edited cells are all dead and they are floating in the sort of soup that we keep the cells in. But the edited cells are alive and they are attached to the surface, the plastic surface of the container that we grow the cells in. And you can also see here that the media on top is a lot more pink, and the media on the bottom is slightly more yellow. That's because there's a pH indicator in it. And as cells metabolize nutrients, they decrease the pH of their environment. So you can see that these cells are happy, they're metabolizing, they are producing all of those metabolites that are turning the media a different color. Whereas up here, the media is just as pink as when it went in because all of the cells have died. So now that we had our proof of concept, we wanted to repeat this methodology in more disease-relevant cells, because this isn't a kidney disease, this is a skin disease. So we repeated this antibiotic resistance experiment in human skin cells, and we got significantly lower rates of cell survival in that antibiotic. So the cells were sort of surviving in small patches rather than in large sheets, which suggests that the editing efficiency is a bit lower, but it is still happening. So we then are our next experiment was to try and integrate the COL7A1 gene instead of the antibiotic resistance gene, because obviously that's the gene that we're really interested in delivering to these patients. And we performed a technique that's called a Western blot, which is a huge pain in the butt, but unfortunately really, really good for telling you what you need to know in this case, which is how much of a specific protein a population of cells is producing. And it shows up, your protein of interest will show up as sort of a black smudge on this membrane. So you can see here, I've included all my controls. So your COL7A1 mutant skin cells that we know for a fact are not producing any collagen 7, there's no smudge. In healthy skin cells, so our positive control, we do see a smudge, which we expect. And unfortunately, in our edited skin cells, we saw no rescue of protein production whatsoever. So it seems like the editing efficiency is too low to actually have a therapeutic effect. So this brings us to step 3 of the research project, which is just what is happening and why isn't this working? So we needed to figure out what exactly is causing this editing efficiency to be so low. And the first thing we wanted to check is this DNA editing machinery actually getting into the cells? And to figure that out, we did what's called tagging of the DNA editing machinery with a molecule that serves as the genetic instructions for the cell to turn green. It's GFP, or green fluorescent protein. It's exactly what it sounds like. It glows green. So with our kidney cells on the right, the green signal is editing machinery that has successfully made it into the cells. And you can see that a lot of these kidney cells are actually turning green. Whereas in the untreated condition, obviously, they are not turning green. Whereas in the skin cells, between the untreated and the edited cells, they look pretty much the same. There is almost no green showing up anywhere in these skin cells, which tells us that there's something going on in the delivery step of trying to get this machinery into these cells. So when we took a closer look at the skin cells that did go green, we essentially isolated the green ones and then looked at their DNA to see what kind of editing had occurred. So only 2% of skin cells internalized the editing machinery, but of those 2% of skin cells, the ones that received that editing machinery, 17% of them were actually successfully edited. And depending on how much you know about DNA editing, this is actually pretty good for the technology that we're using. And it's also known that I believe between 15 and 20% genetic correction of COL7A1 can have a functional therapeutic effect on patients or in patient cells. So this could theoretically, if we actually got it into all of these cells, be a really good basis for a treatment. We just have to figure out how to get it in. So the next thing we did was try to figure out if these cells would internalize other genetic material. So is it something unique about this DNA editing machinery? And it seems as though it is. We tested 7 different formulations of nanoparticles to deliver the genetic material to these skin cells. And in this case, we were delivering just the GFP gene, not the DNA editing machinery plus the GFP gene. And all of the formulations we tested achieved over 60% of green cells. So we know now that it's not necessarily that these cells won't take up any genetic material. It's something specific about this machinery. So the conclusions of this stage of my research have been that the DNA editing machinery does function to a certain extent in both kidney cells and skin cells, but the skin cells, for some reason, are resistant to internalizing it. Despite the fact that other genetic material is readily internalized and expressed by the skin cells. So it undergoes that transcription and translation that I talked about earlier. So the questions we're left with is the DNA editing machinery just not getting internalized, or is it getting internalized but not getting transcribed and translated? And if so, why? And the why question is one that when you're troubleshooting this research, when you're in that sort of what is happening stage, you run up against a lot. And as a researcher, most of your time is spent trying stuff that doesn't work. And this is kind of what I would like everybody to take away from this talk. Is not necessarily anything specific about the data that I've gotten, but just learning a bit about how this kind of research goes. It's a whole lot of what. You spend a lot of time doing stuff that just doesn't work. And when it doesn't work, you have 2 options. You can stay in that what phase and do more troubleshooting, or you can give up and try something new. And giving up sounds like it wouldn't be the best option for patients, but there's a bit of a sunk cost fallacy there, where sometimes a technology, especially a novel technology that has just come out, the year before, still needs to be optimized a little bit before it's actually going to be helpful and applicable to real human beings versus cells in a dish. So sometimes the best thing to do for patients is to give up on a technology that isn't working and move on to the next option. I hope that's more of an optimistic tone than a pessimistic one that we're taking into the Q&A. But if anybody has any questions about my project or about DNA editing in general, please pipe up and let me know.
Right. Thank you, Karina, for that great talk, great introduction as well about the genetic aspects of basically genetic diseases and then gene therapy as well. So I can see we have one question from the chat now. So I'm sorry, I'm going to probably pronounce the name wrong. Mural Goris Amir is asking, will it be challenging to implement CRISPR for genetic diseases? Like, what is your view in terms of CRISPR or other editing approaches in terms of overcoming regulatory?
Yeah, so that's a great question. The good news is some of it already has. So I talked about sickle cell anemia briefly. There is a CRISPR-based therapy for sickle cell anemia that has received FDA approval and is already on the market. And there are several more CRISPR-based therapies that are sort of coming down the pipeline in terms of clinical trials. The difficulty is that CRISPR isn't just one technology, but that's also sort of an advantage, I guess, because the different flavors of CRISPR have different advantages and disadvantages to them that make them very easily tailored to specific diseases. So, for example, with EB, like I talked about, there's a lot of different mutations that cause it. So you need to use a technology that's capable of performing more large-scale edits and potentially replace the entire gene. Whereas with genetic diseases that are caused by just a small handful of mutations, you can use those sort of more nitty-gritty individual mutation corrections. In terms of regulation, one of the big pushes right now is sort of a one gene, one approval approach. So previously, the way that CRISPR works is you have to target it with a specific guide molecule. And that guide molecule is unique to the mutation you're trying to correct. So again, if you have a lot of mutations that are causing a different disease, then you have to, in theory, get clinical approval for all of those different guide molecules. And what's a push that's happening right now is sort of walking back a little bit of that safety regulation and saying that if we can show that correcting a mutation within this gene cures the disease, then we shouldn't have to go through the entire pipeline for each individual guide molecule. We should just sort of be able to say, we've tested this guide in cells, we know that it works, we know that it's safe, and it should be able to sort of join in a pool of approved therapies instead. I hope that sort of clarifies things.
Yep, makes sense. In terms of that, do you have an opinion? Because lately we have had some developments in basically this N of one, like a single patient treatment approaches. So I think the last 2 months there were a few papers describing just a single patient approach, like what you said, a single disease, a single treatment. So do you think these papers will start adding up to this pool of pre-approved therapies?
I certainly hope so. I think that this is a really great, this uptick we're seeing in N of one therapy proof of concept, I guess. I shouldn't call it proof of concept because it is in a real patient, and that has a lot of gravity to it and shouldn't be taken lightly. But I think this is fantastic news for rare disease, especially where there aren't that many patients and maybe even enough patients to do a full clinical trial. And also in patients where we know that these diseases are almost guaranteed to be lethal very, very early in life. And so, again, there's a lot of gravity that needs to be applied to accelerating that pipeline of trying different therapies on real human patients. But especially in the case of rare disease, where it's unlikely that a full clinical trial will be considered cost effective, CRISPR is these sort of bespoke precision medicine therapies could be really game changing for a lot of young patients.
Thank you for the answer. I have another question here from the public. So, Sparky is asking, have you considered using machine learning or dataset splicing for predictive modeling, such as perhaps some runs, such as why perhaps some runs are better than others?
So that's a great question, and I have no idea. If anything, I'm kicking myself for not having learned more about AI and machine learning when I was in school. But obviously, with the boom of that happening right now, there, I think, is going to be a lot more instances of machine learning accelerating these kinds of experiments, especially when it comes to sort of parsing out the human genome, which is this unimaginably complex beast that we know a lot about, and we're pretty sure we know enough about to do certain experiments safely and develop certain therapies very safely. But this sort of lab on a chip approach is going to accelerate that safety testing even more. So I don't know a whole lot about the specifics, but I know that there are a lot of people who are looking into that.
Yes, I think we all more biologist people are wondering how
Exactly.
AI
Yeah.
gonna hopefully help us, or maybe confuse us a little bit as well.
Yeah. I mean, it's definitely confusing to me, but the important part is that there are scientists who are not confused by it. And as far as I'm concerned, I'm happy to let them lead the way on this.
Yeah. I'm just waiting for someone to tell me this works, so maybe now you can apply it to your favorite gene.
Exactly. Yeah. I'm like, just tell me what to put in my cells and I will do it.
Yeah.
Yeah. Or maybe I go back to school. Who knows? Maybe we'll all just need to go get another PhD.
I'll relearn how to
It was easy enough the first time, right?
Oh, a few years, but well.
Yeah, that's fine.
So here we have another question. Elena T. What do you think it is about the passage machinery that makes it either not get into the cells or not express as well compared to just having the GFP gene in there?
So that's the question. That's what we have to decide if we're going to devote a whole bunch of time to figuring out. The sort of prevailing theory we have right now is that it's just a really big piece of machinery. And to figure out how much of an issue that would actually be a pretty straightforward experiment. We just need to essentially make our GFP plasmid a lot bigger. So put multiple copies within the same plasmid so that it sort of has the same physical size. But we're also having trouble getting this to work with what's called electroporation, which is a totally different approach physiologically. So it's difficult to say at this stage whether it's just not going in or whether it's not going in or whether it is going in and just not getting expressed properly. And again, the amount of time that you devote as a researcher to figuring out that more like basic research aspect of the nitty gritty of like what pathways are involved and what can we, why isn't this working versus this tech isn't working, let's move to something else because we want to help these patients is something that every researcher just kind of has to deal with over the course of their career. So yeah, great question. That's the question. And I don't know if we'll ever have an answer because we might just move on to something else.
Yeah, definitely understandable. Then we have Christopher Fordham. So is it is passage something that could be used for many different types of disease if it worked? I'm presuming that's
Absolutely.
yes.
Yeah. That's what's so exciting about this technology to me and technologies like it. So Passage isn't the only technology that could potentially be an option for this whole gene replacement. But yeah, in theory, any monogenic disease, meaning any disease that's caused by one gene, where just putting in a healthy copy of the gene can compensate for, or any, I should say, any recessive monogenic disease, where putting in a healthy copy of the gene can fix the problem. These technologies should work. It's just, will they work? But in theory, yeah, this could be the foundation of a cure for hundreds and hundreds of diseases.
We can only hope that
Yeah,
someday it will work.
yeah, fingers crossed.
Christopher again just wanted to say he said cool glasses and thank you for
Thank
your
you.
work. Then we have Rafael Yanez. So excellent talk, Karina. Very clear and honest, and really liked the analogy to the IKEA model. The question is, why did you choose this particular type of genome editing out of all the options that we currently have?
So it was a little bit of essentially their Passage is very similar to a technology called Paste that was sort of published the year before, but Passage, due to that evolution of that protein machinery, was reported to be 16 times more effective than its precursor. And I, as someone who has a big background in CRISPR specifically, this is a CRISPR-based technology that appeared to be, in its initial reporting, really, really effective. And in its initial reporting, they actually achieved editing in skin cells, which is very difficult. So we thought, based off of its efficacy, its safety, and the fact that they tested it out in a model that we were interested in using, that was what we wanted to go with. And so that's sort of why we chose that as the full gene replacement technology. And then as far as why we chose a full gene replacement technology, again, it has to do with the fact that there are so many different mutations that cause this disease. So if we could just do one and done, one size fits all, that would be really, really good news for patients.
And make everything faster as well.
Yeah.
Instead of trying to go through 800 different variations.
Oh my gosh. Yeah. Well, and see, that's where AI would probably help out a lot, is sort of that screening step of figuring out which guide molecules are predicted to be best. But we were hoping that we wouldn't have to do that.
Hopefully you don't.
Yeah.
Then we have Yarista Pestano Tome. So wanted to thank you for sharing your research. Mm-hmm. And then wanted to ask if all of the 108 genes known mutated, are related to the same protein. I think this is more to the disease that you're researching.
Yeah. So,
and then, so basically, if all of the 108 mutated genes are related to the same protein, and if those genes are coding genes.
So my specific disease just looks at one gene. I'm sorry, I'm confused by the question a bit. If you mean all of these different mutations within that gene are related to the same protein, yes. This collagen 7 or Col7A1 codes for one protein, collagen 7. And if they're in coding regions of the gene, some of them are and some of them aren't. So if I can, hang on, I can pull up my slides again and show you. Here we go. So this image here, on the left side, these are what are called splicing mutations. So they're not necessarily within the protein coding parts of the gene, but they dictate how that DNA gets printed out into mRNA. So if there's a splicing mutation, maybe a page of that instruction manual that gets printed out goes missing. or one gets printed upside down, or however you want to imagine it. And then on the right, these are missense and nonsense mutations. So where parts will get swapped out for one another, or the instructions manual just sort of ends early and you're left with like half of a protein. So coding versus non-coding, there are mutations in both of those categories, which again is part of what makes it so difficult to come up with all these individual guide molecules to fix them all individually. I hope that answers your question.
Yeah, I think that was the question.
Okay, cool.
At least I think that's how I understood it as well.
Okay.
I don't know if I don't think we have more questions from the public. I don't know if Sonam has another question for you.
Thank you, Karina. That was really great. I really enjoyed it.
Thank you.
I actually had the question more on the disease itself. Is there any not therapy necessarily, but I guess at the moment, how are the patients being managed with how terrible it is for them with
skin? So prior to the last 2 years, I would say it was pretty much just medicated bandages and trying to keep infection under control. And I mean, as you might imagine, these patients have a very, very low quality of life. And the squamous cell carcinoma tends to develop and become fatal before the age of 40, I think. And then obviously in areas of the world with less developed healthcare systems, it can be fatal in a matter of infancy. In the past couple of years, though, a couple really exciting developments have come out of the therapy field scene for this. One of them is called BVEC, which is essentially they take, it's a different form of gene therapy. It's not gene editing, it's additive gene therapy. So they take a virus. So remember, viruses are really good at putting their DNA into our cells. And they just take a healthy copy of the gene. And this is really cool because it's topically applied. So it's literally in the form of a cream. And there's viral particles in the cream. You put the cream on the skin and they introduce a healthy copy of the gene. And it works very well. However, because it's non-integrating, it doesn't edit the underlying DNA, you have to keep applying it. And it is insanely expensive. Like the size of a credit card, I think, is thousands and thousands and thousands of dollars. So over the course of a patient's life, it's just prohibitively expensive. More recently, a small molecule therapy based off of birch bark extract was approved. I don't know specifically the molecular interaction that helps with, but apparently it does, which is very exciting and presumably a lot cheaper. And then most recently, I think within the past month or so, scientists have performed and I think have achieved some form of regulatory approval for an autologous cell sheet therapy, where they'll take cells out of a patient, again, use a virus to put in the gene, grow them into these big sheets of essentially skin in a dish, and then surgically suture that fresh skin over the wounds. And that's been very, very promising as well. So it's kind of therapy-wise, the last couple of years have been really exciting for this disease.
Oh, cool. And I had one last question, which you kind of touched upon, is there's always a question of how much of the protein do you actually need to kind of not necessarily cure, but relieve the symptoms of the disease. And you mentioned about 16 to 17% of the protein could potentially be enough. Is that something?
Yeah. So one of the other sort of wrinkles in our strategy that I didn't really get into, so this is what's called a secreted protein. It doesn't do its job within the cell. Its job is to leave the cell and grab onto stuff outside of the cell. And so if you imagine this as like two sheets of fabric that you're trying to keep stuck together, a couple stitches here and there, if they're really strong, can help keep them stuck together. You don't necessarily need the entire thing to be fully woven. So cells that are cranking out functional protein can potentially compensate for neighboring cells that haven't actually received the edit. And so as part of our approach, we're not just putting in the gene, we're putting in the gene a sort of like, you could think of it as like a hyperactive version of the gene, where the promoter that tells the cell when and how much of this gene to turn on is just saying like, go, 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 make as much as you can, because the cell next to you may not have received this edit. It's not necessarily one-to-one between the number of cells that are corrected and the therapeutic effect. You can get away with correcting fewer cells, and they'll compensate for each other.
Yeah. Okay, that's very cool strategy. Yeah. Actually, she replied saying, "Yeah, we answered the question. It was
perfect, great."
I don't think there are any more questions online.
Yeah, I was just going to ask if, well, you're clearly just starting on the research, but I was going to ask if you had thought, how would you apply your therapy if or when it worked, considering that your target area is quite big?
Yes, yeah. So another side of our lab's research, so not my project, but some of the other students, some of the students in the lab, is topical nanoparticles. So similar to the therapy I described, where it's just a cream you put on your skin, but it would be the nanoparticles that we have shown work pretty well and can edit 60% of these cells in cream form. And it's going okay. It turns out your skin has spent many, many tens of thousands of years evolving to do one job, which is to keep stuff out of your body. And so it's pretty difficult to make something that can just sort of slide right through it. But we're working on it. And then another approach that we're thinking of using is essentially, it's recently been shown that the blister fluid from these patients contains stem cells in it. So rather than having to harvest stem cells from a very invasive place like bone marrow, you can just take a syringe, pop a blister, extract the fluid, and you've got some stem cells. And if you can edit the DNA of those stem cells and then inject them back in, it's been shown that that can actually help the cells produce some collagen 7. So that's, I think, what we're more leaning towards now that the nanoparticle stuff has proven to be less easy than we thought. Again, sometimes stuff just doesn't work. And the best thing you can do for patients in the long run is stop banging your head against the wall and figure out the next thing to try.
Perfect. Yeah, I don't think we have any more questions. Let me just check the chat. Yeah, I think that from the public, at least, we don't have any more questions.
Okay, cool.
Sonam, do you have any last questions on your end?
No, not from my end, other than it was really great talk, and I really enjoyed your explanations.
Oh, thank you.
Yeah, I have to say the same. I just love your analogies and how you explain everything.
Please use it. I feel like there's a lot of distrust in science in the world right now, and part of that maybe comes down to us not always knowing how to explain stuff. So if anything I've said you think might help people understand, please go out there and use it.
Yeah, great analogies, great way of explaining everything. So all I have to say from representing the community right now is just thank you for coming today, for explaining to us so well what are you trying to develop, how is it going, even if it's not going so well. And yeah, thank you very much for taking the time to explain to us your job and your work.
Yeah, thank you for having me, and thank you to everybody in the audience for coming and listening.
Yes. I think with that, we're going to end the talk, and I will just like to thank everyone for tuning in today. And hopefully, if you're interested in gene therapy or cell therapy, you will register for our next talk that should be in about 2 or 3 months. We're going to advertise in social media again, as we did today. So thank you very much, everyone, for coming today, and have a great rest of your evening.